Hello everyone, I'm Zhao. I'll be presenting the work Inpainting Cropped Diffusion MRI using deep generative models on behalf of the first author Rafi, who unfortunately could not be here today. The problem we want to tackle here is that in the image acquisition stage of the diffusion-weighted imaging, parts of the brain can be cut off from the field of view due to improper positioning of the subject. This situation often happens in longitudinal studies for investigating neurodevelopment during childhood or adolescence, because the acquisition protocols are often optimized for the baseline visits of the subjects, but then become suboptimal for the increased head size of the subjects in later visits. This cropping artifact not only results in misinformation, but also influences subsequent processing steps such as inter-subject or cross-modality registrations, both of which heavily rely on the image boundary information. The resulting misalignments can affect the follow-up group analysis based on regional DTM measures. We propose to fix the cropping artifact by inpainting the missing part of the brain. Specifically, assuming we have a training set of cropped and full image pairs, we propose to learn a vector context VAE model that can translate the cropped image to the inpainted full image. The VQ-VAE has shown to be able to produce high-quality images compared to the vanilla VAE. The VQ-VAE model has a standard encoder-decoder structure that embeds the input image to a latent space and then reconstructs the latent embedding to the output image. The only difference between VQ-VAE and the vanilla VAE is that instead of formulating a continuous distribution of the latent embeddings, VQ-VAE encourages the latent space to be discrete. That is, the latent embeddings can only take categorical values. Therefore, the training of VQ-VAE not only uses a regular reconstruction loss, but also optimizes a codebook such that each latent embedding is committed to one of the discrete code from the codebook. We further improve the VQ-VAE by using the skip connections motivated by the UNEC structure. In this way, the encoded features at each scale are concatenated to the corresponding decoded features to enable sharp reconstruction. To validate this model, we first performed synthetic experiments that used the diffusion-weighted images of 824 subjects from the Incanda study without cropping artifacts. We synthetically cropped each image by removing the top 8 slices of the image, and then we applied the model to get an inpainted version of the image and compare it to the original full image, which is regarded as the ground truth. We compared a variety of similarity metrics among our proposed model and baselines, including the regular VQ-VAE, UNET, and VAE GAN. Results indicate that our proposed model is potentially a better model for image inpainting. Specifically, the left 4 columns show the similarity metrics between the inpainted images and the ground truth in the whole image space, and the right 4 columns show the similarity only measured in the cropped region. So our model achieved the highest score in most of the similarity metrics. Moreover, these improvements were statistically significant compared to the baselines. The only exception is that the reconstructed image by our proposed model is less sharp than the ones generated by the VAE GAN model according to the gradient measure. But when inspected visually, we found that the VAE GAN method tends to generate noisier images, which might explain the higher gradient measures compared to the proposed model. Moreover, we can see that the baseline VQ-VAE without the skip connections tends to generate over-smooth reconstructions, whereas our proposed model that incorporates the skip connections can generate sharp reconstructions. And finally, we applied the proposed model to real data, which include 13 subjects that were labeled as unusable by the Incanda quality assurance protocol due to the cropping artifact. We inpainted these images by our proposed model, but this time we don't have ground truths anymore, so we cannot do a direct comparison. Instead, we inspected the impact of inpainting on the follow-up registration procedures. Specifically, we first derived the FA map from the original diffusion-weighted image, and then registered that map to the SRI-24 atlas. This alignment was potentially corrupted at the top of the skull due to the loss of boundary information from the cropping. Next, we repeated this registration using the inpainted diffusion-weighted images. Then we can compare the two resulting FA maps. We specifically measured FA difference in the supplementary motor area, which is the region closest to the top of the skull. The right figure shows the difference at various axial slices. We can see that while the two registrations resulted in similar FA values in lower axial slices and also outside the brain, the registration based on inpainted images generated lower FA at slices most severely impacted by the cropping. Note that higher FA estimates are generally associated with greater noise or larger artifacts within the data. So this indicates that our results could potentially improve the power of group analysis on the FA measures. In conclusion, we show that our model can inpaint missing regions in diffusion-weighted images. This inpainting can reduce errors in the data processing pipeline, so we don't necessarily need to discard images with cropping artifacts. This will in turn improve the power of the follow-up statistical analysis. Thanks for your attention.